Thank you so much for tuning in. My name is Jacob Ewald. I am a graduate student in Dr. Christopher Ivey's lab at California State University, Chico, and I am studying the species boundaries in two Northern California monkeyflowers. Now I'm going to start with some background. I'm going to explain what species boundaries are, and we're going to look at a couple of examples to illustrate this concept, and then we'll dive into my project. So at the heart of what I'm studying is the maintenance of species boundaries. That is, how do two closely related species limit gene flow between them? Species boundaries are so important to our understanding of evolution that the biological species concept actually defines species as closely related groups that no longer have any gene flow occurring between them. Now, this gene flow is usually blocked by one or more reproductive barriers that work in concert. Now, these reproductive barriers generally fall into one of two categories. Either they prevent two parent species from producing a hybrid, or they limit the reproductive fitness of a hybrid after it has formed. So pre-hybridization reproductive barriers include things in plants, include things like non-overlapping geographic range, differences in pollinators, differences in flowering times, things of that nature. A great example of these pre-hybridization reproductive barriers occur in two species of California jewel flowers. Now, these two species, they broadly overlap in geographic range. So if I were to take a map, I could point to something and say, okay, obviously these two species overlap in range. But at the fine spatial scale, things start to look quite different. So you see the two jewel flowers are actually isolated based on their soil adaptations. So much so that individuals of the two different species are rarely any closer than 200 meters apart. That's quite a long distance for pollinators to fly. In fact, that pollinator behavior is the final reproductive barrier that isolates these species. So while they share certain pollinators between these two species, those pollinators tend to visit the two species at different rates as well as at different times. So we can understand this example conceptually and see how these barriers work additively. So that fine scale spatial separation limits upwards of 60% of their potential gene flow, and then that the difference in pollinator behavior finishes the job, eliminating the remaining gene flow. On the other hand, there are reproductive barriers that act on the hybrid itself, limiting that hybrid's ability to reproduce. Now, you might be thinking, well, if hybrids are already around, doesn't that mean gene flow has occurred? And it's not quite that simple. You see, if a hybrid dies without reproducing, then any gene flow dies with the hybrid. If there's to be gene flow between two species, what has to happen is you've got two species, they hybridize, they produce this hybrid, but then that hybrid itself has to be able to back-cross, right, with one or the other species. If it cannot back-cross, then no novel genetic information can travel from one species through the hybrid into the other species. A great example of this comes from a 2013 study on yellow monkeyflowers, Mimulus guttatus. And essentially, what we see here is that they generally should be able to reproduce between populations, right? Two populations of the same species normally reproduce just fine. However, there is a unique copper-tolerant population of Mimulus guttatus growing on an old mine in Northern California. This copper-tolerant population, when crossed with non-tolerant populations, produces hybrids that do not survive to flowering. They start growing normally, but they die before any pollen can be produced or any seeds can be produced, therefore eliminating any gene flow. So what this would look like conceptually is those pre-hybridization barriers actually do quite little to limit gene flow, and of course their spatial overlap is quite complete. However, that really strong reproductive barrier provided by the lethal hybrids completely eliminates any potential gene flow. So that brings me to my project. I am interested in the currently unclear species boundaries separating the yellow monkeyflower Mimulus guttatus with the shield-bracted monkeyflower Mimulus glaucescens. So Mimulus guttatus is found across Western North America. It's widespread. Mimulus glaucescens is restricted to Northern California. Now, in this range, in this region of overlap, they actually can grow quite close to each other. They also share pollinators. There's no difference in pollinators. There's no difference in flowering times. And in fact, I've been able to grow greenhouse grown hybrids in my lab, no problem. However, we don't seem to find any hybrids in nature, and we're not sure why. So 2 previous graduate students in Dr. Ivy's lab examined 14 different reproductive barriers between these 2 species. Despite all of that, they were only able to explain a total of about 67% reduction in gene flow. That still leaves quite a lot of gene flow that is unexplained. So that's where I come in. There's one of 2 explanations for what's going on here. The first potential explanation is there are indeed hybrids in nature and we're just missing them. If they're going undetected, then we need to figure out what they look like. So I am examining and measuring traits on these 2 species' most distinctive feature, and that is their bracts. Bracts are these leaf-like structures that grow just beneath the flowering bodies. To understand what these bracts look like and really get a good sense of what they, how they're different, I'm examining their shape and their trichome density. You can think of trichomes basically as tiny hairs. So Mimulus glaucescens gets its common name, shield-bracted monkeyflower, from the nice circular bracts that it has. Mimulus guttatus, on the other hand, has much more oval-shaped bracts. In addition, Mimulus guttatus is quite hairy. Mimulus glaucescens is generally considered completely hairless. The other explanation for what's going on is maybe there is an additional, as yet undescribed, reproductive barrier separating these 2 species. If that's the case, then once we figure out what that is, we can potentially explain the 100% reduction in gene flow. To test that, I'm thinking maybe there's a microhabitat barrier just like those jewel flowers we saw before. So I'm going to be conducting reciprocal transplants between the 2. Essentially, I'm going to be uprooting and planting individuals from one species into their opposite species habitat. Those are going to be my experimental group. And then we're going to do some controls, basically uprooting and replanting the species back into their original habitat. If those native species survive to flowering at a higher rate than the immigrant species, that is a sign that microhabitat is playing some sort of reproductive barrier here. So I do have some results from the shape data, which I just completed. So when measuring bract length over bract width, or dividing bract length and bract width, I am able to get an idea of how circular each bract is. Essentially, the closer the bract length is to the width, the closer the number is going to be to 1, and that means a more circular bract. Clearly, those shield-bracted monkeyflowers, Mimulus glaucescens, are much more circular, but there is a region of overlap at those extreme ends. Trichome density, those hairs are a little clearer. Mimulus guttatus are very hairy as expected. Mimulus glaucescens are almost always entirely bald. So there's still some takeaways and some ambiguities here from the data that I have, and that is, so they do exhibit distinct shapes and distinct hairiness between these 2 species, but would I be able to notice that in the field? And that's where things get a little tricky. Unless I've got, a microscope or a field microscope or magnifying glass, it's going to be hard to tell how hairy something is, let's say. In addition to that, are hybrids, would they produce some sort of intermediate trait, or would they have sort of a Mendelian inheritance pattern, something like a dominant recessive pattern? So that's where I'm going next. I've gone ahead and I've completed all my measurements of the parent species, but I cross-pollinated hybrids, and I'm going to be measuring them next. Following that, I will be growing a 3rd generation, doing some hybrid crosses, as well as some back crosses of hybrids with their parent species, and measuring them as well on these same traits. So ultimately, this is important because even though speciation is a crucial step in that evolutionary process, there's still quite a bit we don't understand about it. In addition to that, the genus Mimulus, these monkeyflowers, are really important for studying speciation because they're so diverse and there's so much we can learn from their diverse genome. In addition to that, Mimulus guttatus and Mimulus glaucesens provide a very rare, unclear set of species boundaries that I'm going to elucidate and further our understanding of speciation. Thank you so much.